Hello, my name is Jessica Fuller, and I'm here to give my summary on the molecular basis of cancer and what I've learned so far this semester. Cancer is a disease caused when cells divide uncontrollably and spread in surrounding tissues, blood, and lymph systems. Cancers create malignant tumors. These tumors are cancer cells that grow through cell division to multiply and spread into different body organs. Most cancer happens from a virus first. So one of the most common viruses that became cancerous was HPV, which is papillomavirus. It's also a part of the herpes family. This virus you can get it by sex. It's basically a sexually transmitted disease. So you can get it by having unprotected sex or anything sexually you're not being protected. So one thing that's most common about this cancer is that it creates warts. So you see warts on herpes 1 and herpes 2, but these warts can become cancerous because they are forming on your skin and they can spread all around your body, and the cells can go through a mutation where they become cancerous and causes cancer. A tumor is basically a neoplasia, which is a new growth of malignant tumor. Differential cells. Malignant tumors are consisting of differential cells, high growth rates, capable of metastasis, invasive, and carcinoma in situ. And a regular tumor would be a differential cells, slower growth rate, encapsulated, no metastasis, no invasive, can cause harm by putting pressure on surrounding tissues because of the tumor growing, but it's at slow growth rate. Benign tumors are easily able to catch and be able to remove depending on where it is located, but it can be cancerous if you do not catch it in time. If you're not knowing your body, you're not catching it in time. Most cancer is created into stages when you are diagnosing it to a patient. Stage 1 is no lymph nodes, no metastasis. It's about 1 inch of a tumor. Stage 2 is contacting the local lymph nodes and no metastasis, but it's about 2.5 centimeters. Stage 3 is nodal involvement and no metastasis, and it's about the same, 2.5 centimeters. And stage 4, and also is metastasis and nodal involvement, and it's over 5 centimeters of the tumor. The tumor is over 5 centimeters, and once it's at metastasis, it has spread it to different parts of the body where you have the main tumor in the primary location where it may start in the lung or the cancer. That's the primary tumor, and once it has spread it to the brain or the breast or the lung or wherever it starts after it, those are secondary tumors. Common cases of cancers are breast, lung, and colorectal. Cancer is either lymphomal, sarcoidal, or carcinoma. Lymphoma begins in lymphocytes and attacks the T cells or B cells. Sarcoidal, it begins in bone and soft tissues, including the muscle and blood and fat. Carcinoma is basically the epithelial cells outside and inside the body. One thing about cancer cells is they create cell death, targeting our normal cells that go through cell division and have proper function to regulate. One thing a tumor lacks is proper function. So once our normal cells go through cell division on a regular basis, they go through cell division. Once they are going through those cycles and those checkpoints to make sure that nothing is going wrong, some things can go wrong, such as DNA damage that can target the DNA sequence and create a mutation, which can offset a oncogene. So some of the DNA damages are single-stranded DNA breaks, damaged bases, mismatched bases, deamination of cytosine. So those are the main common ones that basically the cell goes through and targets those breaks and tries to repair it before it goes through mutation. So most of the DNA repair pathways catch it before it gets to a the end of cell division where it multiplies and spreads. So some of the DNA repair pathways are base excision repair, mismatch repair, nucleotide excision repair, homologous recombination repair, non-homologous end joining, and translesion DNA synthesis. Another way that our body can help us from getting those cancer cells to stop and not spread before it gets to that level is cellular senescence. Once the body knows that they see an abnormal cell going through cell division and it's keep going, the cell stops its propagation of damaged or defective cells. It stops the propagation of those cells. The only way it can continue and break that cellular senescence is if it's an oncogene that's very strong, and or the cancer cells signal the proteins or signal the genes that are normal genes to basically say that it's a healthy gene or it's a gene that's here to help you. In reality, it's a gene that's not here to help you. It's here to cause damage to your cells. So once a cancer cell is in your body and it starts going through cell division, it goes 3 different ways to target those normal cells to create cell death. Some of these ways are apoptosis, necrosis, and autophagy. Apoptosis is cell shrinkage, membrane blebs, and chromatin condensation. Necrosis is cell swelling, inflammation, rupture of membranes, and loss of organelle structure. Autophagy is cell digestion, formation of large lysosomal vacuoles. One thing that we have to also help us with targeting those cancer genes once they are dividing is our tumor suppressant genes. The tumor suppressant genes targets the oncogenes that are in our cells right now that are dividing. It acts as negative regulators of the oncogenes. So tumor suppressant genes encode proteins that are functional integrated into pathways that prevent unscheduled cell proliferation, stimulate death, or trigger the induction of permanent cell cycle arrest. One of the common tumor suppressant genes are p53. It controls the cell growth and division, but this gene can also switch and be helpful to the cancer cells. So that's one thing about tumor suppressant genes. They can switch on and off because of the cancer cells signaling the proteins to target those genes, to switch the genes to help the tumor grow, and say that it's a good gene. It can help you. And when in reality, it's a bad gene and it's killing your cells. Also, other tumor suppressant genes are BRCA2, which is in breast and ovarian cancer, and that targets DNA repair. So once you have breast cancer, that's one of the genes that targets the DNA, that targets those your DNA sequence so that it won't create a mutation. The oncogenes are genes that encode proteins and are capable of inducing cell transformation. Oncogenes are basic genes that can be mutated and cause cancer. Become cancerous. Sorry about that. They act as overexpression of activating mutations. They interact with normal cellular proteins involving in cell growth and proliferation. And the cancer malignant properties are in the cells of the oncogenes. Once a cancer cell is creating a tumor, the tumor has to survive in the environment that it's in. It has to grow. One thing a scientist has stated in 1889, Stephen Paget, he did an autopsy experiment on 735 breast cancer patients that had metastasis in 4 different tissues: lung, liver, uterus, and bone. And so he was wondering why metastasis has spread into different environments. So he created the seed and cell hypothesis, and basically he was wondering why they spread into different environments and why they were surviving and proliferating in those environments. And one thing a tumor has to be in is a microenvironment in order for it to survive. It has to be in their own environment for it to exceed and grow. So his hypothesis was seed and soil hypothesis, and it stated every single cancer cell must be regarded as an organism alive and capable of development. When a plant goes to seed, its seeds are carried in all directions, but they can only live and grow if they fall on oncogenic soil. So basically what that means, if an oncogene is known, such as HER2 is known for being in breast cancer, that's one of the cells that are found in breast cancer, as well as BRCA2, 1 and 2. But HER2 is known in breast cancer, and you wouldn't see HER2 in lung cancer because it wouldn't be able to grow, it wouldn't be able to proliferate or go through cell division. You would see it in breast cancer. But one thing you can notice is that those genes can spread during metastasis. It can spread, and those will be considered the secondary tumors because it has started in breast cancer, it has moved its way through other body organs by basically angiogenesis, metastases, creating vessels, tumor vessels to basically provide oxygen and nutrients to survive. One thing a tumor vessel are exposed to is the acidic microenvironment that is characterized by oxygen and nutrient deprivation. So cancer cells, in a sense, tumor cancer cells create an environment that they create their own glucose, their own ATP, their own oxygen to survive in those environments. And they do that, one of the major sources they do that for ATP and energy is glycolysis. That's the major source they do to survive, the cancer cells survive, proliferate, and cell divide. They also go through they get their nutrients from the regulated growth factors, and they also go through TCA cycle and PI3K pathway that activates mutations of RTK, which is a kinase. One of the things I've stated with angiogenesis is that it is providing blood vessels, it's creating blood vessels contacting blood vessels to other body organs for it so it can spread. And it's doing that by endothelial cells from existing vessels. It responds to the oncogenic signals by degrading the basement membrane, loosening association with parasites and smooth muscles, and altering their morphology and proliferating. And so some of the most common cancers go through that environment where some of the most common cancers or tumors are in a microenvironment where they're surviving. Like again, tumor lacks proper function. So the tumor microenvironment consists of the extracellular matrix, the microfibroblasts, adipose cells, immune inflammatory cells, blood and lymphatic vascular networks. Some of the ways that scientists and researchers and doctors are targeting these cancer cells is through radiation, chemotherapy, and surgery. So most patients either go through surgery depending on what stage of cancer they're on. And one way to identify what stage of cancer they're on is through the TNM test. The TNM test is the T standing for measuring the size of the tumor, the N is the lymph node involvement, and the M is the metastasis where it's at. So you decide what stage you're on depending on this test and what your results are getting and what cancer cells are found, what genes are found in the area they're testing. So chemotherapy is targeting of medications that they use to target the cancer cells, which can cause also other side effects depending on what medications they're on. Radiation is just low gamma rays that can target the cancer cells that are killing them through cell death. And of course, surgery is just removing the tumor and hoping that it doesn't arise or come back. So most patients, when they remove the tumor, they go through either chemotherapy or radiation, or they go through both, depending on what stage you're on. So I definitely know that technology has advanced a lot from the 1900s and the 1800s, when cancer was just evolving and we found about what it was coming from and how we tested mice to identify what ways we can target those biomarkers of cancer of the area they're in, like breast cancer or lung cancer. They target those, chemotherapy targets those biomarkers that can slow down the process so that the process of proliferating and creating a bigger tumor. So technology has advanced a lot, and I definitely know that there is a cure for cancer. The knowledge is out there. It's forever growing. The area is forever changing, and definitely cancer is definitely a silent killer. So I definitely, my grandfather died from cancer and my grandma, so I definitely know the preventative measures that you take to stop cancer or stop it from growing, or prevent it from growing, or identifying your DNA sequence to see if you possess any oncogenes that can be mutated at any type of risk factor. Because one thing about oncogenes, they can mutate off of risk factors such as your lifestyle, your alcohol that you're drinking, smoking, gaining too much weight, overweight, or not active, or just hereditary. So it's still a process on how cancer is started and how and why. So that's still a question that is still getting answered. So I definitely enjoyed learning about this subject a lot. I did a couple different assignments with papers with, I did one researching breast cancer, and I also did one with ovarian cancer. And I enjoyed it a lot. It was a nice course. And thank you for watching. I appreciate it.